Hello, my name is Todd Knepper, and I'm excited to be part of this roundtable discussion talking about practice models in precision oncology, representing Moffit Cancer Center with my colleague Christine Walko. A little bit of background on myself. I actually completed a precision oncology fellowship after graduating from pharmacy school, starting in 2014, and have since stayed on through the development of our program here at Moffit, and really excited to talk to you about it, and I think and kind of show the growth and the development of our program from where we started to where we are now, and some of the unique things about practice at an academic cancer center. So with that, I'd like to welcome my colleague Christine Walko as we talk with you about our model.
Thanks, Todd. It is a pleasure to be with everyone to tell you about our model and how we have incorporated precision medicine into our clinical practice. So as Dr. Knepper said, my name is Chris Walko. I was recruited down to Moffit Cancer Center back in 2014 as well. And then Todd soon became our first fellow. After graduating pharmacy school, I did a pharmacy practice residency at the Medical College of Virginia in Richmond. And then I did my oncology residency and fellowship training at the University of North Carolina and stayed there for several years before coming down to Moffit. So just a brief overview about Moffit Cancer Center. We are a nonprofit standalone cancer center that was actually on the younger side, so established back in 1986. We are located in Tampa, Florida, with multiple locations throughout the Tampa Bay area. We are the only NCCN designated comprehensive cancer center in Florida, and we typically see over 100,000 unique patients per year. This includes more than 31,000 new patients annually, and a big focus of our clinical care is in not just stem cell transplantation, where we do more than 400 a year, but the growing role of CAR T. So this includes both the myeloid, lymphoid, as well as solid tumors, especially in the clinical trial space. And we do more than 2,000 CAR T visits per year. We also have more than 350 enrolling clinical trials. So I just want to introduce you to our team. So our precision medicine clinical service at Moffit includes 4 attendings: Dr. Kevin Hicks, Dr. Teresa Ho, Dr. Todd Knepper, who's with us today, and myself. And then we also have 2 precision medicine associates, Dr. Juliana Baliou and Paige Parkinson. So the way that we have kind of chosen or felt was best to separate out the different cases is, as our team grew from just 3 people to 6 now, it made a lot of sense for us to specialize in certain malignancies. So back in 2014, we kind of covered everything, and we evolved from being personalized medicine. We were initially in the cancer epidemiology department. Over time, they actually created a new department, the Department of Individualized Cancer Management, or DICAM, where we were with the genetic counselors, our gene home for hereditary inherited cancer syndromes. Senior adult was in there, AYA was in there, survivorship was in there, and it kind of made sense at the time. However, as precision medicine really became standard of practice, we then were moved with the genetic counselors into the Department of Pathology. And so we are, our boss is Dr. Aaron Bossler, who is the head of molecular pathology. And that has been a really, really great relationship that we'll talk about a little bit more, and then hopefully even more when we do the discussion part of this. So as you can see from the different faculty, Dr. Kevin Hicks focuses efforts in the thoracic realm. He also does some work in pancreatic, as well as leading a lot of the germline pharmacogenomics and the implementation aspect, which we'll talk a little bit about our database, but having someone who is knowledgeable and passionate about implementation really, really is critical to thinking about how your data is going to be generated, how your data is going to be stored, how your data is going to be accessed, and how you're going to overcome the natural challenges with technology. And so we are very, very fortunate to have Kevin, who has a desire and a passion in that area. Dr. Teresa Ho covers our patients with pancreatic cancer, colorectal cancer, and breast cancer. She also works with Kevin on our germline pharmacogenomic initiatives. Dr. Todd Knepper, you'll hear a little bit more about some of his efforts in the heme and myeloid realm. That's really exciting. I think Moffit is one of the programs that does both heme, lymphoid, and solid tumors for next generation sequencing. Todd also works with our cutaneous patients, and we all do a fair amount of education, but Todd is also our point person and the coordinator of the HemOnc Fellow education. We have our about 9 HemOnc fellows, so our MD fellows, who spend a month with us in their second year as part of their standard fellowship at Moffit. And then I kind of take all of the rare cancers, which I actually really enjoy the challenge of that and seeing how technology has helped us a lot. Neuro-oncology, sarcomas, head, neck, and endocrine, our gyn-onc group, genitourinary, and then I do some of the other GI currently, like cholangiocarcinoma. And then essential to our workflow is having the support of Juliana and Paige. Both of them actually used to work in our clinical trials area and then have come over to work with us, and they support so much of our effort through data coordination, our database management, preparing our lists for molecular tumor boards, helping with clinical trial matching and keeping up with all of that, as well as some collaborative initiatives such as our website. Ultimately, our mission for the Precision Medicine Clinical Service is to seamlessly translate biomarker testing through the patient journey to inform treatment options, including targeted therapies, clinical trial eligibility, and patient safety risk mitigation. And about a year and a half ago, we all met to kind of form this vision as well as map out our strategic themes. And so the 4 strategic themes that we focus our efforts around include establishing best-in-class dissemination of information. This includes integrating our biomarker reporting with therapy and clinical trial identification, as well as a lot of our cross-functional partnerships, enhancing external engagements. We do have partnerships with outside institutions, including Memorial Hospital on the eastern side of Florida. Positively impacting the patient experience. So really considering the full patient journey for a patient who is seen at Moffit and where precision medicine can have an impact. And also our patient safety monitoring, so our pharmacogenomic initiatives, which are not the focus of this talk, but that are, there has been a lot going on in that space, and Kevin and Teresa have done a phenomenal job of leading those initiatives. We also aim to streamline our clinical trial identification, so making sure that if a trial is available and enrolling for a patient who we are reviewing the next generation sequencing for, that we try to identify that, as well as thinking about opportunities for clinical trials that we may not have, but that we have a patient population who could benefit. So we work very closely with our early therapeutics team. So our testing and ordering model, this kind of flow diagram basically just shows how we really are the ferry that crosses the chasm of interpreting the biomarker testing results and turning them into a clinical action that is hopefully valuable to the patient. So we take all of the next generation sequencing reports, and so these are ordered by the treating team. We are not typically involved with that. However, we will provide guidance if asked, but basically we get involved whenever a test result comes back into our workflow. And so we do next generation sequencing through both an in-house assay. It is we call it the Moffit STAR assay. It is a DNA RNA based 500 gene panel that's on the Illumina platform that may be similar to something that you may have at your institution. I know other institutions do utilize that same kind of platform. We also will interpret commercial assays, so Foundation Medicine and Guardant are the 2 big ones that we interpret, but any patient who has next generation sequencing done on any platform, even from the outside, the team can consult us and we will review it. We will then take that data as it comes in, and we'll talk about that in the next slide, but we'll put it together with the other molecular results. So there may be IHC results, FISH results, methylomic results, and then also, very importantly, putting it in context with the patient's narrative. So looking at the patient's clinical history, their goals of care, and then obviously the pathology, the imaging, everything to kind of make that story, we will take into consideration and then formulate a precision medicine clinical consult. And we'll show you a little bit of what that looks like. And what we're really looking for are on-label therapy options, off-label therapy options, clinical trial options, prognostic evidence. So sometimes with both the heme and lymphoid panels, as well as the rarer malignancies, so the sarcomas, we may actually be just kind of looking for is the alteration actionable? Is it helping us to kind of understand what histology we may be looking at? So it may be questions that are prognostic and not just predictive. We also look for risk mitigation and treatment optimization, as well as identification of potential germline findings. And so in order to process all of this, each of us are in charge of our cancers that we take care of and synthesizing all that data to create a report. However, we definitely do not work alone. We have created multiple molecular tumor boards, kind of with different goals in mind. Whenever we first started back in 2014, 2015, we had one big molecular tumor board that we called the Clinical Genomic Action Committee, and we would review all types of cases, heme, lymphoid, solid tumor, and we would have a very, very diverse team of individuals who would attend these meetings. As next generation sequencing really became much more standard and people became a lot more comfortable with it, plus meeting loads and things like that became higher, we really felt that we needed to have kind of a different solution. And so we have a few different types of molecular tumor boards at Moffit. We have one molecular tumor board, a multidisciplinary molecular tumor board that Dr. Knepper runs once a week that focuses on malignant hematology. So our myeloid and lymphoid patients are discussed there along with medical hematology and molecular pathology, the heme path molecular pathologists. We have a solid tumor molecular tumor board includes our team, molecular pathology, as well as one of our genetic counselors to really look at kind of some rare tumors or rare alterations in a more common tumor to kind of get a consensus. This is oftentimes where we'll dig deeper into an odd fusion or a weird splice site, or if we have a cancer of unknown primary, to kind of have everyone take a look at it and think, does it look like something? What other testing could we potentially recommend? So it's really a working meeting. Dr. Kevin Hicks chairs a thoracic molecular tumor board that is heavily focused on reviewing all of the cases that come in who are seen in the thoracic clinic, who have next generation sequencing, and matching them to the clinical trials. We have numerous clinical trials, and obviously there's numerous biomarkers in that space. And so he chairs that molecular tumor board with the goal of really helping to bridge the gap to identify both current clinical trials as well as future clinical trials that patients may go on. I co-chair a gynecologic oncology molecular tumor board monthly, where the focus is heavily on education, and I work with the gyn-onc fellow who will discuss the cases. We do highlight the clinical trials that patients may go on, but we also will highlight unique biomarkers. In this group, microsatellite status instability, or MSI high, homologous recombination deficiency often come up, and kind of digging into those and the different methods of detecting it is oftentimes a discussion there. And then finally, we also do have a partnership with Memorial Hospital, and we have a molecular tumor board that we work on with them. And with that, I'm going to turn it over to Dr. Knepper to talk to you about how we ingest our data and utilize our data, as well as store our data.
Yeah, thanks so much. I'm glad to be back. So as Dr. Walko mentioned, the tests that we review come from a variety of different sources, whether it be internal tests for solid tumors, internal tests for hematologic malignancies, of which we have several now, one geared towards myeloid malignancies, one geared towards lymphoid malignancies, and then all of the external commercial tests that physicians can order as well. So as you might imagine, all those reports are going to look differently, come in differently. So we needed to develop tools to standardize those across the board so that our notes look the same, and then all that data gets ingested and collected in a way that supports the institutional goals for data integration. So one tool that we have for that is our precision medicine data mart. Started back when we really started here at Moffit with our genomic database that we kind of got going. It was somewhat rudimentary back in the day, but allowed us to ingest that information, and really it was created in the sense of helping us to put together our clinical notes, so supporting the clinical aims of what we were doing. So kind of starting with that as our starting point, it's since built over time. As you can see, the volumes of cases that we see have really increased over time as precision medicine has been integrated into more and more cancer types. More therapies have been developed across different malignancies, and more clinical trials are now available that are enrolling patients with on the basis of molecular features that are detected in their tumor. So now, in a somewhat more spread out utilitarian way, the internal NGS tests as well as the external tests, when they're ordered through the electronic health record, we receive that information back and through a machine readable file, which kind of populates a broader data lake, and then through our MCAP, through our precision medicine data mart, communicating with the MCAP platform, the Moffit Cancer Analytics platform, we're able to essentially flag that information, say we're looking for this MRN medical record number, and then a specific test identification number, and that pulls that information for us to do our review and conduct our notes. So it's able to identify this unique test and then populate for us the specific mutations that are detected, so what gene, the variant allele frequency, and additional kind of basic information about the test, like what's the name of the test, what's the date that the test is reported back, additional specimen information, all information that's discreetly available, is discreetly input on the testing report, we're able to pull and populate to kind of get us started with our clinical notes. And then, as you can see in the figure, the connection with an interpretative database that we're kind of able to manually build when it comes to specific alterations that we may see more frequently. This way, when we see it again, we have kind of a starting point for our interpretation, and we're able to take that, integrate it with the individual case in front of us, and provide a targeted review and summary and interpretation / recommendation, in many ways I can say it, to the treatment team in the form of a consult note, which, as you can see there, is a semi annotated note, and then entering that into electronic health records. So when our notes go in, they're essentially going to look the same, whether it's our internal report or the external reports. Of course, those original NGS reports that are, if they're in-house, are created by our excellent molecular pathology team, or the external PDFs are available for the treatment team as well. But our work takes that additional level, right? So when you're getting those NGS reports back, the testing companies, even the internal tests, are not necessarily going to know additional information about the patient. They're not going to know their previous treatment. They're not going to know the goals of treatment. Those clinical variables that we're able to understand as being part of the treatment team and look up and kind of provide that more precise, more personalized interpretation of the results to bridge that gap and really to help the treatment team to decide what treatment decision, like what treatments are potentially available based on these results, and what the interpretation of the results can mean for broader therapeutic decision making and just better understanding of the patient's disease. Yeah, I think we can, so that probably hits everything for that slide, so we can move on to the next slide. Yeah, so this is a little bit more technical, but just kind of showing the integration of the molecular data with the broader cloud-based data infrastructure at the institution. Given we kind of started doing that initial organization of the data back when we first came in and kind of started our program here, and that in a lot of ways had formed the foundation for which the broader data infrastructure was built. So we had all of this information available and kind of ingesting that and building out the broader data cloud infrastructure on the basis of the molecular data and building it on top of that. So being able to connect several different data sources into a broader platform that now kind of creates a bit of a loop where the information now comes into the MCAP system, and we're able to extract out specific information that we need into our own data mart that we're able to use to build our precision medicine consults. The next slide, I think, is good. Yeah, so here's a bit of a view of what that semi-automated consult might look like, and some of the things that can kind of get pre-populated for us. So some of this information gets automatically extracted from the NGS report, like I mentioned, the specific mutations that are reported, the test performed, the date, and some information. And we have the ability, when we're entering this into the database, to kind of manually edit this as well. So the diagnosis, the prior therapy, if we wanted to add that, are in there, and then a pre-populated interpretation kind of to get us started. So like the most recent version of the interpretation that takes the information from at the gene mutation level, so a specific mutation within a gene, and connecting that to the diagnosis itself. So in this example here, it's giving a specific interpretation that's tied to KRAS G12C in lung cancer. If it was a different KRAS mutation, it might look a little bit different. If it was a different cancer type, it would look different as well. So being able to kind of hit those kind of three touch points: diagnosis, gene, mutation, and triggering the start of this automated consult node, and kind of just gets us started with at least one of the interpretation. And it's going to vary by the results of the test. If there's kind of multiple alterations that get reported back that have interpretations on them, they'll populate. If there's only one, but then there's additional alterations that are of potential clinical significance that are worthy of discussing in the note, we would just add that to our kind of the note as we build it before it gets finalized, and include that in the broader summary. I think that's it. Yeah. So then continuing on. So I think the kind of a question here is asking, so like how we're consulted and kind of access to us. And the way that it works, I think, as we've talked about, there's probably two big buckets, I guess you would say, of the tests that come back. So there's our internal NGS results and tests that gets ordered, and then the external results as well. As you might imagine, with our internal results and ordering process, there's a bit more customization that we can layer on top of it. So our treatment teams, when they're ordering the NGS results, ordering the NGS tests, have the opportunity right up front to request that a member of our team provides a consultation. So essentially, what that means is enters a note on that report. So that's for our internal tests, and we'll do that. And then for the external commercial tests, we're essentially reviewing those automatically, in a sense. You'll have like discussion with our providers in those clinical areas in terms of their expectations and their preferences for whether or not we're going to be entering notes for all of the cases, and just kind of building that relationship and having that discussion, I think, is also something that's key in terms of communicating with your providers in terms of what they want, what will help them to support their practice, and to make sure that they're properly interpreting the NGS results and properly utilizing the NGS results to support patient care. I think a lot of ways we talk about ourselves as essentially a new subspecialty. So like the molecular oncology subspecialty, where we have an analogous to the way a radiologist is has expertise in interpretation of X-rays and CT scans and complex imaging results, and provides that interpretation to the ordering physician and pathologist. Will do the same in terms of the complex laboratory tests that get ordered. They have the interpretate, then they do the review and give an interpretation to the treating team. Our role here as precision oncology specialists, I think, is analogous to that, where we have these complex tests that get ordered, and we're able to subspecialize in the interpretation of these results as pharmacists, having expertise in the therapies that are available, having awareness of the clinical trials that are ongoing and potentially in development, and being able to connect our expertise as pharmacists, our experience and expertise in precision oncology and genomics, and tying that all together. So, and then kind of for the next slide, obviously it's not just us, it's a broader team that have to come together to support precision oncology. And I think this is just a list of some of the partners from genetics, for example. These complex next generation sequencing tests, they're intended to detect somatic mutations, but there are alterations that get reported back that could potentially be germline alterations. So having access to and partnership with a team of genetic counselors, medical geneticists who specialize in the interpretation of the genetic results, and kind of knowing if and when definitive germline testing is needed to be ordered is really an essential partner. Of course, pathology as well. So as Dr. Walko mentioned, our team has currently found a home in the Department of Pathology, molecular pathology in particular, which I think has really been a really good match for the work that we do, the way that we think about cases, the role that we play, and part of the broader treatment team. And we still continue to work with them as our close partners on our molecular tumor boards, this pathology representation all the time. Of course, the patient care team who are ordering the tests, and then as we talked about our databases, the information technology, the IT teams, and as well as bioinformatics and medical informatics partners to help to build these databases, support these databases, make sure this information is getting ingested properly, and then kind of the outputs on the same way. And I think just kind of broadly speaking, precision oncology, we've come a long way in terms of the therapies that are now FDA approved and available for the patients on the basis of the results of next generation sequencing tests. But clinical trials are really still a critical part of these results as well. So making sure that we have partners and awareness and knowledge of the ongoing clinical trials that are available at our institution. As an academic cancer center, we're fortunate that we do have many clinical trials that are available to our patients here, including those that are enrolling patients on the basis of specific biomarkers that are detected by the test. But then potentially looking beyond our walls for that as well. So this is a little bit of an aside here. I think from our experience, we've been here at Moffit for over a decade at this point, and I think that's really helped us, being integrated into the institution, being seen as the precision oncology experts, and being able to interpret these NGS results and kind of translating that into clinical significance. So understanding what this a particular finding means, not just kind of at the functional and biological level, but the clinical level, and translating that into patient care. And kind of once we've established ourselves as at the institution, we've started to kind of get looked at to expand into new initiatives and to kind of meet the needs as they arise across the institute. So from that, just wanted to highlight a couple areas where our precision medicine team has been integrated and essentially taking a leadership role alongside of our other providers in pathology and in hematologic malignancies or medical oncology as well. So a couple of examples here. So recognizing kind of a challenge of the interpretation of next generation sequencing results for patients after transplant for myeloid malignancies, we were asked to review those results and to help with the interpretation of the findings that come back. So we developed a process for doing that and built a structured electronic health record documentation for our results to help differentiate kind of different interpretations of the alterations that come back, that range from something that's totally benign to something that might require intervention. And when people who are maybe not as accustomed to reviewing these results see next generation sequencing reports, there could be a question about what's the functional significance, what's the clinical significance of these results. So as our team has developed this experience and expertise with the interpretation of next generation sequencing tests, it became kind of a natural. We were a natural team for other providers to look for providing additional support. And then another aspect that I wanted to highlight as well is a specialized precursor-focused clinic that we've stood up here at our institute, the CATCH Clinic. It's Counseling and Therapeutics for Clonal Hematopoiesis, where partnering ourselves, malignant hematology, pathology, cardio-oncology, partnering, and to try and identify clonal hematopoiesis and identify patients who would potentially benefit from kind of that specialized assessment and management in the clinic. So as a function of that, our team had helped to kind of start it and stand it up from the beginning, including developing specific order sets in electronic health record with partners from clinical informatics, and then again developing a standardized process for which to review those results and then deliver the interpretation of those findings to the ordering provider as well. So I think those are just kind of a couple of examples that once we've established our team as precision medicine experts at the institute, just kind of meeting the evolving needs of the field as things continue to grow and develop.
Thanks, Todd. And so just to quickly wrap up, I think as you're thinking about how to incorporate precision medicine into your institution, really considering your own institutional needs, the technology that is available, and just understanding that patient care standards are going to change over time. There is a constant need to really reevaluate and innovate based on the changing environment. Where we were when we started back in 2014 and 2015 really laid the foundation for where we are now, but we have new technology, we have new challenges, we're going to be getting a new electronic health system. All of these things change, and I think you really have to keep changing with it and thinking about what is becoming more standard of care and where can you continue to layer in and really contribute to the value of patient care as well as the goals of the institution. And I think one of the other things that we've really learned is that one molecular tumor board model may not fit best everywhere. We have tumor histology agnostic molecular tumor boards, both for solid tumor and in the heme realm. We have cancer specific tumor boards, the thoracic molecular tumor board that Dr. Hicks leads, the gynonc molecular tumor board that I work with, and then we have educational focused molecular tumor boards as well, which is our a little bit more like our memorial molecular tumor board. And honestly, education and communication helps us to continue to evolve, which is why I'm so happy to participate in this effort and look forward to the roundtable discussion.